the RH blood group system. So the RH blood group system versus the ABO group. So the antigen structure and locations, we're going to talk about that first. So the RH antigens, these reside on proteins embedded in the red blood cell membrane. They are not glycosylated, so they're not carbohydrates. Where the ABO group, they are found on the carbohydrate chains, and they are attached to lipids and proteins on the red blood cell surface. For immunogenicity, so the Rh antigens, these are highly immunogenic. They are capable of triggering a strong immune response. They are particularly important in transfusion medicine and pregnancy, which can cause hemolytic disease of the newborn. For antibody production, the Rh antibodies, they are not naturally occurring. They are produced after exposure to foreign red blood cells through either blood transfusion or fetal-maternal hemorrhage. This is typically an IgG class antibody, which can cross the placenta. So the importance of the Rh. So the Rh is the second most important blood group system after ABO. It plays a major role in transfusion medicine and pregnancy management. For the complexity of it, so the Rh system includes over 50 distinct red blood cell antigens, and the most clinically significant antigen is the Rhd, or Rh negative or positive. So we have Rhd status, so individuals who possess the Rhd antigen are classified as Rh positive. Individuals who lack the Rhd or just D antigen are Rh negative. For clinical relevance, so determining the Rhd status is essential during pre-transfusion testing to avoid alloimmunization and during prenatal screening to prevent hemolytic disease of the fetus and newborn. So the discovery of the Rh blood group system. So Levine and Stetson back in 1939 described a hemolytic transfusion reaction in an OB patient. This reaction suggested the presence of an unknown blood group antigen. So back in 1940, then we have Landsteiner and Weiner, and they injected guinea pigs and rabbits with red blood cells from the rhesus macaque monkeys. The animals produced an antibody that reacted with human red blood cells. This led to the identification of the Rh factor, though the antigen they identified was not identical to human Rhd, but the name Rh still remained and actually came from the rhesus monkey, so Rh and rhesus. So some further research helped establish that the Rh antigen is a primary cause of hemolytic disease of the fetus and newborn, also known as erythroblastosis fetalis. This occurs when an Rh negative mother produces antibodies against Rh positive fetal red blood cells. For Rh and transfusion reactions, so Rh incompatibility is a major cause of hemolytic transfusion reactions. This occurs when the Rh negative individuals are infused or transfused with Rh positive blood, and this can result in immune-mediated red blood cell destruction. Then we have the genetics for the Rh antigen. So Fisher-Race theory. The Fisher-Race theory proposed that the Rh system antigens are encoded by three closely linked sets of alleles, commonly referred to as D, big C, little c, and big E, little e. Each gene is responsible for producing a specific antigen on the red blood cell surface. So just as kind of some important notes, so we have the combination of alleles inherited will determine the Rh antigen profile, and this model helps explain the inheritance patterns of the Rh antigens in populations. Each Rh antigen and its corresponding gene share the same letter designation. So when referring to the gene, the letter is italicized, like big D, little c, big E, something like that. They're all italicized. When referring to the antigen, the letter is capitalized and non-italicized. For the phenotype definition, the Rh phenotype is a red blood cell, is determined by the presence or absence of 5 main antigens, and those are big D, big C, little c, big E, and little e. These are identified through serological typing. The majority, or about 85% of Caucasians are Rh positive, which means they have the D antigen present. Little e antigen is the most common at 98%, while big E antigen is the least common at 30%. So then we have Weiner's hypothesis. So he proposed a single gene model for the Rh system. According to this model, one Rh gene produces a single agglutinogen, so it's a complex antigen. This agglutinogen contains multiple blood factors, at least three distinct different components. For key distinction for the Fisher-Race, so Fisher-Race model suggests three closely linked genes. The Wiener model suggests one gene with multiple alleles that produce multi-factor agglutinogens. Just a little side note, so despite different genetic interpretations, both models do describe the same serologic antigens, which is that big D, big C, big E, little e. For immunologic expression, the agglutinogen as a phenotype, so the agglutinogen, which is that complex antigen, is a phenotypic expression of a haplotype. A haplotype, just as a reminder, is a set of genes inherited together from a single parent. For the antigenic composition, so each factor within the agglutinogen is an antigen that can recognize by a specific antibody and can be targeted individually or as a group by antibodies. So these antibodies may recognize a single Rh factor, such as anti-E, or recognize multiple factors within the agglutinogen, so depending on the immune response and the antigenic structure. The Landsteiner-Wiener blood group. So this system was identified around the same time as the Rh antigens when they were first discovered. The antibody associated with the Landsteiner-Wiener system was initially produced by injecting rhesus monkey red blood cells into guinea pigs and rabbits. This antibody was found to have the same specificity as an antibody described earlier by Levine and Stetson, linking Landsteiner-Wiener to Rh in terms of historical discovery. Later research showed that the antibodies described were not identical. The anti-rhesus antibody originally described by Landsteiner and Wiener was renamed anti-LW in their honor. There is a phenotypic similarity between the Rh and LW blood groups systems, though. So anti-Landsteiner-Wiener reacts strongly with most D-positive red blood cells. It reacts weakly or sometimes not at all with the Rh-negative red blood cells. It never reacts with the Rh null cells and usually will show stronger positive reactions with D-positive red blood cells compared to D-negative adult red blood cells. These react equally well with cord cells regardless of their D-type. Then we had the Rosenfield nomenclature system, so some background on this. So in the early 1960s, Rosenfield and Associates developed a new system for Rh antigen identification. The goal was to create a numerical and standardized way to document Rh antigen presence or absence and replace confusing genetic nomenclatures. So some system features for Rosenfield was each Rh antigen is assigned a number based on order of discovery or its recognized relationship to the Rh system. This system helped to provide a phenotypic representation. It does not infer genotype, and it only shows whether a specific antigen is present or absent on the red blood cell surface. If there's a minus sign before a number, it indicates the absence of that antigen. Antigens are not typed, are not included in the sequence. So for example, a phenotype showing presence of big D and absence of big C, big E, and little e would be written as an Rh of 1, -2, -3, -4, -5. The ISBT, or the International Society of Blood Transfusion, formed the Committee on Terminology for Red Cell Surface Antigen. The committee's mandate was to establish a uniform nomenclature system that would help ensure the nomenclature is and machine readable and to help align nomenclature with the genetic basis of blood groups. The ISBT adopted a 6-digit number for each authenticated antigen within a blood group system. The numbering system structure, how this works, is the first 3 digits represent the blood group system. The last 3 digits indicate the specific antigen within that system. So the Rh blood group system was assigned the number 004. So each Rh antigen was given a unique number following 004 to complete the 6-digit identifier. So Fisher-Race identifies the antigens by letter, so by that D, big C, big E, little e. Wiener uses the capital R and little r with the superscripts, 0 or A, and that helps designate the haplotypes. Rosenfield uses the RH that's followed by a number to indicate presence of specific antigen, and then ISBT uses that 6-digit number, so 004 for RH system, plus the 3 digits for antigen. So this is just a little overview of what we just talked about, nice on a little, spreadsheet there for you. For consistency of use, the RHD and RHCE, all uppercase and all in italics, will be used from this point forward in the text to help indicate genes. So we have the RHD, RH big C little e, RH little c big E, RH little c little e, and RH big C big E. So these will be used to designate proteins on which the RH antigens reside. So then we have molecular genetics, so the theories of Rh genetic control. Two main theories were initially proposed to explain genetic results from serologic and biochemical studies in the Rh system. For Wiener's theory, a single gene produces a single product. This product contains multiple separately recognizable factors. Where Fisher and Race's theory, they had the Rh locus contains 3 distinct genes, and each gene controls the production of its respective antigen. Tippett's proposal. So Tippett proposed that 2 closely linked genes located on chromosome 1 control the expression of the Rh proteins. So the RHD gene encodes for the presence or absence of the RHD protein, whereas RHCE gene codes for one of the following polypeptides. So either the RH big C little e, little c big E, little c little e, or big C big E. So the Rh-associated glycoprotein, or the RAG gene, is located on the chromosome 6. It produces the Rh-associated glycoprotein. RAG acts as a co-expressor and is essential for the successful expression of Rh antigens on the red blood cell surface. So the RAG gene is located on chromosome 6. It produces the Rh-associated glycoprotein. As mentioned before, it acts as a co-expressor, and it is essential for the successful expression of Rh antigens on the blood cell surface. By itself, RAG does not express any Rh antigens. Mutations in the RAG gene can cause missing or significantly altered RHD and Rh big C big E proteins, affecting antigen expression. Just an important note, the Rh null phenotype results from such mutations that can affect the Rh antigen expression. Rh positive individuals inherit 1 or 2 codominant Rh big D genes, which lead to the expression of the RHD antigen and results in an Rh positive blood type. In addition to the RHD genes, 2 Rh big C big E genes are inherited, one from each parent. These Rh big C big E genes encode for the Rh big C big E proteins, contributing to the overall Rh antigen profile of an individual. The Rh negative status can arise due to the absence or alteration of the RHD gene or antigen on the red blood cell. This Rh negative phenotype can result from at least 3 different genetic mutations affecting the RHD gene or its expression. These mutations vary in type and frequency and are strongly associated with ethnic background. So with European ethnicity, the most common cause is a complete deletion of the RHD gene, leading to no production of that RHD protein. If there's African ethnicity, the mutations often include RHD pseudogenes, which is a non-functional gene variant, or altered forms of the gene resulting in reduced or altered expression of RHD antigen. For Asian ethnicity, so there's a common mutation that include hybrid genes, where portions of the RHD and Rh big C big E genes are combined, leading to altered RHD antigen expression or weak expression. These differences do have important implications for blood typing, for transfusion compatibility, and understanding the population genetics. So Rh antigens are non-glycosylated proteins, meaning they do not have a carbohydrate group attached. These antigens are located on transmembrane proteins that span the red blood cell membrane, making them an integral part of the cell's structure. Only small extracellular loops of these Rh proteins are exposed on the surface of the red blood cell, which are the portions recognized by antibodies. The density of D antigen sites on red blood cells will vary depending on the individual's Rh phenotype. So individuals with the Rh positive phenotype tend to have a higher number of D antigen sites. The exact number of D antigen sites can vary widely among individuals and can influence the strength of antibody reactions. The RhD and Rh big C big E proteins, along with the reg, are found exclusively on red blood cells. As transmembrane proteins, they span the red blood cell membrane and contribute to the cell's structural integrity, helping maintain the shape and stability of the red blood cell. Beyond structural roles, these proteins may also serve as molecular transporters, facilitating the movement of ions or small molecules across the red blood cell membrane. Their exclusive presence on red blood cells makes them critical markers in the blood typing and transfusion medicine. So weak D phenotypes. So some individuals express a weaker form of the D antigen on their red blood cells, which may not be detectable by routine serologic testing. To detect this weaker expression, the indirect antiglobulin test, or the IAT, is often required. Variants of the D antigen with altered expression are grouped into three main phenotypes. So we have weak D, which is characterized by a reduced quantity of the D antigen on the red blood cell surface. These individuals typically have a complete but a diminished D antigen. Partial D, these are individuals that express only parts of the D antigen due to changes in the protein structure, and they may develop anti-D antibodies if they are exposed to the missing parts of the antigen through either transfusion or pregnancy. Then we have the D sub superscript EL. So this is where the D antigen is present in extremely low amounts. It's often undetectable except by absorption elution techniques. This is common in some Asian population. So once again, recognition of these phenotypes is critical for transfusion safety and Rh immunoprophylaxis, as some individuals with weak or partial D types may be at risk of alloimmunization. Most Rh antibodies are of the IgG class, which means they react best at body temperature, which is 37 degrees Celsius, or during the antiglobulin phase, after the addition of the anti-human globulin during that testing. These antibodies are typically produced following exposure to foreign Rh antigens, such as through blood transfusion or pregnancy with an Rh positive fetus. Rh antibodies often will show a dosage effect, meaning they react more strongly with red blood cells that are homozygous for the antigen compared to the heterozygous cells. Testing for Rh antibodies is often enhanced by using enzyme-treated red blood cells, such as papain or fisin, which can increase the sensitivity of the test by modifying the red blood cell surface and exposing or enhancing antigen sites. These characteristics make Rh antibodies clinically significant, as they can cause hemolytic transfusion reactions and hemolytic disease of the fetus and newborn. Rh antibodies often persist for many years after initial sensitization. Once formed, these antibodies can remain detectable in the circulation indefinitely, making it important to consider past transfusions or pregnancies when evaluating antibody presence. Unlike some other blood group antibodies, Rh antibodies do not typically bind complement, which means they usually cause hemolysis through immune-mediated destruction rather than complement-mediated lysis. Rh antibodies are capable of crossing the placenta, which allows them to bind to fetal red blood cells that express the corresponding Rh antigen. These transplacental transfer can lead to hemolytic disease of the fetus in newborn, which is a serious condition where fetal red blood cells are actually destroyed. The goal in Rh typing is to use a reagent anti-D antibody that enables rapid and accurate determination of the individual's RhD status, and similar in ease and precision to the ABO typing. For different types of reagents are available to achieve this goal, including high protein-based reagents. These contain a higher concentration of proteins to promote stronger agglutination reaction. A low protein-based reagent, this is designed to reduce false positive caused by non-specific reactions, often preferred for improved specificity. We also have saline-based reagents. These use a saline as a medium and are usually milder, reducing non-specific reactions. We also have chemically modified reagents. So these are altered antibodies or chemical modifications to improve stability and reactivity. And then we also have monoclonal antibodies. These are highly specific uniform antibodies produced by a single clone, offering consistent and reproducible results. And for the last one, we have a blend of monoclonals. This actually combines multiple monoclonal antibodies to cover various epitopes on the D antigen, improving detection of the variant D phenotypes. So the D antigen is the most immunogenic antigen outside of the ABO blood group system, meaning it is highly likely to stimulate an immune response when exposed to foreign red blood cells. So when assessing for possible immune reactions involving the D antigen, careful review of the patient's medical history is essential. And that would include history of pregnancy. So a fetal maternal hemorrhage can expose maternal immune system to the D antigen. We're going to look at their history of transfusion of blood products containing red blood cells, which could also introduce foreign D antigens. Some clinical signs that may suggest an immune reaction include an unexplained fever, mild bilirubin elevation indicating that there is possible hemolysis, or a decrease in hemoglobin and haptoglobin, which is going to be consistent with destruction of red blood cells. For laboratory testing, we may do a direct antiglobulin test, or a DAT, and this will usually be positive at this time, which would indicate antibody or complement bound to red blood cells. Antibody screen may reveal a circulating anti-D antibody in the patient's serum, and elution studies can help identify antibodies bound to the patient's red blood cells, and this is useful in complex cases. For hemolytic disease of the fetus in newborns, this is caused by Rh antibodies, and this is tends to be pretty severe because those Rh antigens, particularly the D antigen, are well developed on fetal red blood cells even in early gestation. Maternal IgG Rh antibodies can cross the placenta and bind to fetal red blood cells, expressing the D antigen, which will then lead to their destruction or hemolysis. To prevent Rh alloimmunization in D-negative mothers, Rh immune globulin, or RIG, is administered. So RIG is a purified preparation of IgG anti-D antibodies. It is given to D-negative women during pregnancy, generally around the 28-week gestation mark, and after delivery if the newborn is Rh positive. So the mechanism of RIG and kind of how it works is it binds any fetal Rh positive red blood cells in the maternal circulation, which prevents the mother's immune system from recognizing and then reacting to them. RIG is only effective in preventing Rh positive related hemolytic disease of the fetus and newborn. It does not prevent alloimmunization to other Rh antigens or non-Rh antigens. So the use of RIG has dramatically reduced the incidence of Rhd alloimmunization and Rh hemolytic disease of the fetus and newborn. Then we have the Rh null syndrome. So people who have Rh null syndrome, they lack all Rh antigens on their red blood cells and are said to have Rh null syndrome. This is a rare condition that can lead to abnormal red blood cell shape and function, as Rh proteins contribute to the red blood cell membrane integrity. Rh null individuals may develop mild hemolytic anemia due to the fragile red blood cells. Then we also have the Rh superscript mod phenotype. So Rh mod phenotype will involve partial suppression of Rh gene expression. This is typically caused by mutations in the RAG gene, which encodes for the Rh-associated glycoprotein. The RAG is essential for the proper expression of the Rh antigens on the red blood cell surface. So when the RAG protein is altered, the normal Rh antigens, which were the big D, big C, big E, little e, are also altered, often resulting in weakened expression of the Rh and LW antigens. This can affect blood typing and transfusion compatibility.